In this video, you will learn how to purify and obtain RNA according to the Qiagen RNeasy technique. Before beginning, make sure that long hair is secured in a braid or a bun, and put on gloves and a lab coat that has clean sleeves. Spray all surfaces and equipment, including your hands, with RNase DNase-free cleaner. First, vortex the cells on high speed for approximately 1 minute. Verify the volume of RLT that the cells were harvested in. Add an equal volume of ethanol to the cell and RLT suspension. This volume will be either 350 or 600 microliters. To ensure complete mixing, vortex the solution at speed 1 or mix by pipetting. We will now start using an RNeasy spin column. The spin column has a membrane at the bottom and sits inside a 2 milliliter collection tube. Transfer up to 700 microliters of the cell and ethanol suspension into the collection tube, being very careful not to touch the membrane at the bottom. Centrifuge for 30 seconds at 10,000 RPM to pull the solution through the filter. Discard the flow-through into a waste beaker. Tap the column on a Kim wipe to remove the excess droplets from the tube, and then replace the column in the collection tube. Transfer the remaining solution into the spin column and repeat the centrifuge for 30 seconds at 10,000 RPM. Discard the flow-through as previously described. Add 700 microliters of buffer RW1 to the spin column. Centrifuge for 30 seconds at 10,000 RPM and discard the flow-through as previously shown. Now add 500 microliters of buffer RPE to the column. Again, centrifuge for 30 seconds at 10,000 RPM and discard the flow-through. Add an additional 500 microliters of buffer RPE to the column. This time, centrifuge for 2 minutes at 10,000 RPM. Instead of discarding the flow-through, discard the entire collection tube and place the spin column into a clean 2 milliliter collection tube. Centrifuge the column for 1 minute at full speed. After this, take the lid off of the centrifuge and allow the column to air dry for 20 minutes. After the 20 minutes, take the spin column and place it in a 1.5 milliliter collection tube with a lid. Now add 30 microliters of RNase-free molecular biology grade water. Make sure that the lid of the collection tube is in a position where it won't snap off. Then centrifuge the column for 1 minute at 10,000 RPM. Now, discard the spin column and not the collection tube. The RNA sample was eluted with the water. To learn how to quantify RNA, please watch the next series of videos about how to use the nanodrop spectrophotometer.